Chronic fatigue and weakness, dizziness, swollen limbs, sudden excruciating pain in the chest, backs, legs, and arms, uncontrollable painful erections or priapism, tissue necrosis, organ damage, increased susceptibility to bacterial infection, death at a young age from stroke, heart attack, or organ failure. These are but a few of the awful symptoms of sickle cell disease, also known as sickle cell anemia, resulting from a defective gene that causes sufferers' blood cells to collapse into a distinctive crescent shape. This inherited disorder afflicts nearly 8 million people worldwide and kills over 34,000 every year, with most sufferers hailing from or tracing their ancestry to tropical regions such as Sub-Saharan Africa. As a result, sickle cell anemia is often seen as a predominantly black disease, tragically affecting access to care for millions of sufferers. But while this affliction causes untold suffering worldwide, condemning millions to a lifetime of pain and, in many cases, an early grave, there is a silver lining, an unexpected evolutionary advantage that has allowed sickle cell anemia to persist for thousands of years. So join us as we take a deep dive into one of the world's most fascinating, yet overlooked diseases. In the regions where it is endemic, sickle cell disease has been known about for centuries, being a major cause of child mortality. Indeed, among the Igbo people of Nigeria, the disease became closely associated with ogbanjis, literally children who come and go, evil spirits thought to possess children and bring ill fortune upon families. However, it was not until the 1870s that the symptoms came to the attention of Western medicine, and not until the 20th century that it was recognized as a distinct disease. In 1904, one Walter Clement Knoll, a 20-year-old dentistry student from the Caribbean island of Grenada, checked into the Presbyterian Hospital in Chicago. For 3 years, Knoll had been suffering from persistent anemia and bouts of severe limb, chest, and abdominal pain, described in the medical language of the time as muscular rheumatism and bilious attacks. At the Presbyterian, Knoll came under the care of Dr. James B. Herrick, who assigned the case to his resident, Dr. Ernest E. Irons. While examining a sample of Knoll's blood under a microscope, Irons made an unusual discovery. Many of his patients' erythrocytes, or red blood cells responsible for carrying oxygen throughout the body, had collapsed into a distinctive crescent, or sickle shape. Dr. Herrick took an active interest in this previously unknown condition, and in November 1910 published the first monograph on the subject, titled Peculiar Elongated and Sickle-Shaped Red Blood Corpuscles in a Case of Severe Anemia. Walter Knoll received treatment from Herrick for 3 years before graduating university and returning home to Grenada to practice dentistry. He died of pneumonia in 1916 at the age of just 32. Several more cases soon appeared in the medical literature, and in 1922, Vern Mason, a medical resident at Johns Hopkins Hospital in Baltimore, Maryland, connected them all together and gave the unusual syndrome its modern name, sickle cell anemia. Mason also noted that all sufferers identified so far were of African or Caribbean origin, leading to the long-held association between sickle cell anemia and black people. Today, the disease is known to affect people from many regions, including India, the Middle East, and the Mediterranean, including Italy, Greece, and Turkey, the common factor being not race, but climate, and we've got more on that later. But while doctors could now identify sickle cell anemia, for decades nobody knew what caused it, with researchers debating whether it was a standalone disorder or merely a symptom of some other disease. In 1927, researchers Vernon Hahn and Elizabeth Gillespie made an important breakthrough when they discovered that the red blood cells of certain people could be made to collapse or sickle when deprived of oxygen, and revived their original round, smooth shape when said oxygen was restored. While those whose blood exhibited this phenomenon, which Hahn and Gillespie dubbed sickle trait, did not suffer from full-blown sickle cell anemia, their relatives often did, suggesting that the disease was hereditary. In 1930, Jesse Shriver and Theodore Waugh replicated Hahn and Gillespie's findings in vivo, that is, in a living person, by wrapping a rubber band around the finger of a 7-year-old girl suffering from sickle cell anemia. This ligature, which created a stagnant, low-oxygen region in the bloodstream, caused the proportion of sickle cells in the girl's bloodstream to jump from 15 to nearly 95%. However, it was not until the late 1940s that the full picture finally started to come together. In 1949, British military physician Colonel E.A. Beat and University of Michigan geneticist Dr. James V. Neal both published papers theorizing that sickle cell anemia was an autosomal recessive disorder. Those with sickle trait, they argued, were heterozygous carriers, having inherited only one copy of a defective gene from their parents, while those with full-blown sickle cell anemia were homozygous, having inherited 2 copies. Thinking back to high school biology, this is similar to how traits like hair and eye color are inherited. The genes or alleles for brown eyes and hair, for example, are dominant, while those for blue eyes or red hair are recessive. Thus, in order to have blue eyes or red hair, you must inherit 2 copies of the recessive allele from your parents, otherwise the dominant allele will be expressed and your hair and eyes will be brown. At the same time, a newly developed analytical technique known as gel electrophoresis revealed an important clue about the molecular nature of sickle cell anemia. This involves placing a mixture of proteins on a sheet of agarose gel, a jello-like substance derived from seaweed, and subjecting them to a strong electric field. The molecules move at different rates depending on their mass, causing them to segregate themselves by size. When applied to hemoglobin, the protein in red blood cells that helps transport oxygen around the body, gel electrophoresis revealed that hemoglobin from sickle cell sufferers moved at a different rate than those from healthy individuals, suggesting that the disease was caused by a defect in this protein. This theory was confirmed in 1949 by American biochemists Linus Pauling, Harvey Atano, S.J. Singer, and Ibert Wells, working at the California Institute of Technology, who published their findings in a landmark paper titled Sickle Cell Anemia, a Molecular Disease in the journal Science. Sickle cell anemia was the first such molecular disease, today usually known as genetic disorders, to be identified. And today we're aware of hundreds more, including hemophilia, cystic fibrosis, porphyria, muscular dystrophy, and Tay-Sachs disease. As an aside, Linus Pauling later became the only person to win 2 unshared Nobel Prizes, and one of only 2, alongside with Marie Curie, to win in 2 different fields, being awarded the prize for chemistry in 1954 for his work on chemical bonds, and for peace in 1962 for his nuclear disarmament activism. Later in his career, however, he began advocating megadoses of vitamin C for the treatment of all manner of diseases, including the common cold and even cancer, a pseudoscientific claim that seriously damaged his reputation, but that's a story for another video. In 1956, Dr. Vernon Ingram expanded upon the work of Pauling and his colleagues, pinpointing the exact defect in hemoglobin that led to the sickling of red blood cells. At the time, however, modern genetics was in its infancy, James Watson and Francis Crick having only revealed the structure of DNA 3 years before, so it was several decades before the physiopathy of sickle cell anemia, or as it became more generally known, sickle cell disease, or SCD, was worked out. To understand the causes and symptoms of sickle cell disease, it is first necessary to discuss the role played by hemoglobin in the body. While we like to think of oxygen as a benign, life-sustaining element, in reality it is highly reactive and must be carefully handled to prevent it from wreaking havoc on tissues. This is where hemoglobin comes in. Hemoglobin is a large, complex protein composed of 2 intricately folded chains of alpha and beta globin. Each of these chains contains 2 hemogroups, ring-shaped molecules containing iron, which give mammalian blood its distinctive red color. These heme groups bind tightly to oxygen molecules, preventing them from escaping until they are needed for cellular respiration. Each hemoglobin molecule can carry up to 4 oxygen molecules, while red blood cells are 96% hemoglobin by dry mass. Indeed, they are little more than flexible, donut-shaped bags of hemoglobin containing little in the way of organelles, DNA, or other common cellular features. This allows blood to have 70 times the oxygen carrying capacity it otherwise would if the oxygen was simply dissolved in the plasma. Adult red blood cells mostly contain hemoglobin A, but the blood of fetuses and very young children contains other forms of the protein called hemoglobin F and hemoglobin A2. And as a brief aside, other animal phyla use respiratory pigments containing different binding elements, giving their blood different hues. For example, a blue-green copper-based compound called hemocyanin is found in the blood or hemolymph of mollusks like clams or octopus, arthropods like crabs, insects and spiders, and presumably Vulcans like Mr. Spock. Similarly, worms and other invertebrates use different iron-based pigments like hemerythrin, erythrochronin, and chlorochronin, which appear pink or purple, while the blood of some unusual invertebrates called tunicates or sea squirts contain vanadium-based compounds called vanabins. These are the only organism known to naturally accumulate and use this toxic heavy metal. Alright, getting back to mammals, muscle tissue also contains a similar but simpler molecule called myoglobin that helps store oxygen long-term. It is myoglobin, not hemoglobin, that is responsible for the color of red meat and the blood that oozes out of a rare steak. And finally, while heme groups have a great affinity for oxygen, they have an even greater affinity for carbon monoxide and will preferentially bind to it, suffocating the body from the inside out. And that's what makes carbon monoxide poisoning so insidious and deadly. In any event, Getting back to sickle cell anemia, hemoglobin, like all proteins, is composed of long chains of amino acids, 22 varieties of which appear in living organisms. The assembly of amino acids into proteins is directed by sections of genetic code, aka genes coded into our DNA. The classic double helix of DNA, full name deoxyribonucleic acid, comprises 2 strands made up of 4 different kinds of nucleic acids or nucleotides: adenine, thymine, guanine, and cytosine, typically abbreviated as A, T, C, and G. These nucleotides bond across the double helix to form so-called base pairs, with A always bonding with T and C with G. In eukaryotic organisms, like ourselves, the entire genetic code is stored as strands of DNA in structures called chromosomes located in the nucleus of every single cell. When a cell needs to produce a certain protein, be it a structural molecule like collagen, an enzyme, or transport molecule like hemoglobin, the section of DNA containing the genes coding for that protein is unzipped by an enzyme called helicase, producing 2 single strands. Free nucleic acid then binds onto the exposed nucleotides, assembling themselves into a molecule known as messenger ribonucleic acid, or mRNA. Yes, that's the same mRNA used in COVID vaccines. This is effectively a half molecule of DNA, only thymine, or T, is replaced by another nucleic acid called uracil, or U. Once assembled, the mRNA passes through a cellular organelle called a ribosome, which reads the nucleotide sequence coded into the strand and uses it to string together amino acids into the required protein. Each amino acid is represented by 1 or more nucleotide triplets or codons in the RNA strand. For example, UGU and UGC code for cysteine. In healthy hemoglobin, the 6th amino acid position in each beta-globin subunit is normally occupied by glutamic acid, but in people with sickle cell disease, this is instead replaced by valine, producing an abnormal protein known as hemoglobin S. This substitution results from a gene called HBB, located on chromosome 11, specifically the adenine in the GAG codon for glutamic acid is replaced with uracil to form the GUG codon for valine. As previously mentioned, 2 copies of this mutant HBB gene are required in order to develop sickle cell disease. If a person inherits only 1 copy, they will carry the sickle cell trait and can pass it on to their children, but will suffer no symptoms of the disease. Thus, if both parents carry 1 copy, their children will have a 1 in 4 chance of developing the diseases. If one parent has the disease and one is a carrier, this probability increases to 1 in 2, while if both parents have the disease, their children are guaranteed to develop it. Yay, the Punnett squares. While this mutation might seem minor, its effects can be catastrophic. Normal ahemoglobin retains its structure, whether transporting oxygen or not, allowing red blood cells to maintain their shape. But when mutated S-hemoglobin releases its oxygen, its beta chains stick together and polymerize into thick 14-strand bundles. Repeated oxygenation and deoxygenation, as these cells cycle through the body, cause the abnormal hemoglobin to crystallize and the red blood cells to distort and collapse into the rigid crescent shape characteristic of sickle cell disease. This phenomenon affects the body in several ways. Firstly, sickle cells are highly susceptible to bursting or hemolysis, and the destruction of so many red blood cells produces severe anemia, significantly reducing the blood's oxygen-carrying capacity and leading to chronic fatigue, weakness, dizziness, and other symptoms. Usually, the sufferer's bone marrow ramps up red cell production to compensate, but it is rarely able to keep up with the sheer attrition rate. Indeed, while the average half-life of a healthy red blood cell is around 90 to 120 days, in a person with sickle cell disease, this drops down to as few as 10 to 20 days. In children, untreated chronic anemia can lead to stunted growth and delayed mental development. Furthermore, the destruction of red blood cells releases large amounts of free heme into the bloodstream. Not only is heme a powerful oxidant damaging the walls of blood vessels, but it is also an alarmin, a signal of tissue damage and infection. High levels of heme in the bloodstream can thus trigger severe inflammation and further tissue damage and pain. Secondly, unlike healthy red blood cells, which are flexible and can squeeze through even the tiniest capillaries, sickle cells easily get stuck and clumped together, causing all manner of trouble. Typically, the first major symptom of sickle cell disease is to develop dactylitis, a painful swelling of the fingers and toes caused by sickle cells blocking blood flow through capillaries. These symptoms first manifest at around 6 months of age, when a child's bone marrow switches from producing F, or fetal hemoglobin, to A, or in this case, S hemoglobin. Other symptoms soon follow, including painful vaso-occlusive crises, or VOCs, also known as sickling crises. These can take place anywhere in the body, though when the pain primarily affects the lungs, it is typically referred to as acute chest syndrome. While these attacks can take place at any time, they're often triggered by events which produce low oxygen or hypoxic conditions in the body, such as stress, cold, dehydration, heavy exertion, or increases in altitude. Sickle cells can also build up and damage various organs, including the kidneys, heart, and especially the spleen, which normally serves to filter defective cells from the bloodstream. Sickle cells, however, quickly clog up and overwhelm this filter, scarring the spleen and causing it to atrophy. This, in turn, weakens the sufferer's immune system, making them more vulnerable to infections by pathogens like streptococcus and parvovirus. Even more unpleasant, a buildup of sickle cell in men can prevent blood from draining from the penis's erectile tissue, causing prolonged, painful, and potentially dangerous erections or priapism. And finally, sickle cells can potentially block vital blood cells, leading to fatal strokes, heart attacks, and pulmonary embolisms. As a result, sufferers of sickle cell disease have life expectancies far lower than the general population, 40 to 60 years in the developed world and even lower in poorer regions. Indeed, in certain parts of Sub-Saharan Africa, 50 to 90% of children born with the disease die before the age of 5. While sickle cell disease with 2 S-beta-globin subunits is the most common, there are other forms of the disease. For example, in sickle hemoglobin C or HBSC disease, one subunit is replaced with hemoglobin S and another with another mutated form of hemoglobin C. This form of sickle cell disease is typically milder than the more common variant. Another related genetic disorder is thalassemia, which results in deficient production of either alpha or beta globin and related symptoms like severe anemia and deformation of the bones in children as their marrow expands to compensate. The disease takes its name from the Greek thalassa, or the sea, as the first cases were reported in people of Mediterranean origin. Unluckily, it is possible for a person to inherit both the mutations for sickle cell and thalassemia, resulting in a disease called S-beta thalassemia. Sickle cell disease afflicts an estimated 8 million people worldwide, while around 400 million carry the sickle cell trait. The disease kills an estimated 34,000 people per year and contributes significantly to a further 400,000 deaths. Despite this, however, the disease has been around for a very long time, with the HBB gene mutation thought to have emerged somewhere in the Sahara Desert some 7,300 years ago. This presents something of an evolutionary quandary, for given its debilitating and life-shortening effects, one would expect sickle cell disease to have been selected out of the gene pool and disappeared long ago. Yet, it persists, suggesting that even this horrible condition must confer some sort of evolutionary advantage. And as it turns out, it does. Remember how we said most sufferers can trace their ancestry to hot or tropical regions like Sub-Saharan Africa, India, the Middle East, the Mediterranean, and the Caribbean? Well, those areas just happen to be hotspots for a disease that kills nearly 1 million people every year: malaria. And sickle cell anemia just so happens to protect its carriers from that very disease. So to understand how this can be, we first need to understand how malaria spreads and develops. Malaria is caused by single-cell parasites of the genus Plasmodium, of which there are 6 known species which infect humans. These parasites are in turn transmitted through the bites of infected Anopheles mosquitoes. Plasmodium eggs, or oocysts, hatch inside the mosquito's gut, releasing tiny worm-like larvae known as sporozoites. When the mosquito bites a human to score a blood meal, the sporozoites enter their new host's bloodstream and make their way to the liver, where they invade liver cells and multiply into thousands of spherical forms known as merozoites. After 7 to 10 days, these burst out of the liver and spread through the bloodstream, where they seek out and occupy new homes: red blood cells. Of all the cells in the human body, with the possible exception of immune cells like T cells and macrophages, red blood cells are perhaps the perfect place for a parasite to hide. As previously mentioned, red blood cells are little more than flexible bags crammed with hemoglobin. As such, they thus lack most of the organelles and functions found in other cells, including the ability to signal the immune system that they've been invaded. Thus, as soon as Plasmodium slithers its way inside, it effectively becomes invisible, allowing it to drift through the host bloodstream unmolested. As it does, it sets to work transforming the barren red blood cell into a proper home, erecting an internal network of channels and scaffolding to help transport nutrients and wastes in and out of the cell, all the while gorging itself on abundant supplies of hemoglobin. But this clever charade can't last forever. All red blood cells must eventually pass through the spleen, which lets only the youngest, spryest specimens through and digests the rest. But while Plasmodium's internal remodeling renders its host cell less flexible than normal, the parasite is able to deploy a set of molecules called chaperones to fool the spleen into leaving it alone, preventing the infected cells from being destroyed along with their clandestine passengers. Eventually, however, the time comes when even this subterfuge is no longer enough, and Plasmodium instead deploys a set of latches on the exterior of the cell, anchoring it to the wall of a blood vessel and avoiding the spleen altogether. It is these anchored blood cells and the damage they cause to blood vessels which produce many of the classic signs and symptoms of malaria: joint and muscle pain, seizures, coma, and liver and spleen damage. Now, unlike the plain exterior of a normal red blood cell, these latches can be detected by the immune system, which immediately begins assembling an army of antibodies and immune cells to destroy the invader. But by the time this army is ready, the merozoites have already burst out and invaded new red blood cells, starting the whole cycle over again. And when it comes time for this new generation to anchor their cellular shelters, the latches they produce will be slightly different than before, their molecular composition pulled from a large arsenal of genes evolved specifically for this purpose. And so the parasite stays always one step ahead of the host. These breakouts of merozoites from red blood cells occur every 2 to 3 days, triggering another classic symptom of malaria: cyclic bouts of tertian or quartan fever and chills. At some point in their life cycle, the merozoites will develop into sex cells called gametocytes, which mature inside the host's bone marrow before running to the bloodstream to be sucked up by another feeding mosquito. Inside the mosquito's gut, the male and female gametocytes mate and produce oocysts, which hatch into sporozoites, starting the whole cycle over again. So, what does any of this have to do with sickle cell disease? Well, it so happens that the red blood cells of those with sickle cell trait, that is, those who inherited only one copy of the defective HBB gene, are far more difficult for Plasmodium to survive in. They also show greater signs of external damage when infiltrated by the parasite, causing them to be swiftly filtered out and destroyed by the spleen. As a result, in regions where malaria is endemic, those with sickle cell trait are significantly less likely to contract malaria and display far lower parasite counts than those who don't. This effect appears to be greatest in early childhood, bridging the dangerous gap between a child losing the passive immunity acquired from their mother and acquiring active immunity. And while this trait comes with significant risks, namely that of inheriting 2 copies and developing sickle cell disease, in evolutionary terms, this is more than offset by the overall reduction in mortality from malaria, allowing the mutation to survive in the human population for thousands of years. Indeed, so useful was this mutation that genetic evidence suggests it may have emerged independently no less than 4 separate times, 3 times in Africa, known as the Benin, Senegal, and Bantu haplotypes, and once in Arabia or India. Other researchers, however, point to a single origin, with the mutation first emerging in West Africa or the Sahara 7,300 years ago and spreading outwards throughout Sub-Saharan Africa and north and eastward towards southern Europe and India. And if you're currently thinking, wait, there's no malaria in southern Europe, what are you talking about? And you'd be correct, there isn't anymore. Indeed, until recently, the disease was endemic across large areas of Europe, with Oliver Cromwell, victor of the English Civil War and Lord Protector of the Commonwealth of England, dying of the disease in 1658. Allegedly, the staunchly Puritan Cromwell refused to take cinchona bark, the source of the anti-malarial drug quinine, dismissing it as a Jesuit cure. Whatever you say, Ollie, it's your funeral. Literally. And in 1943, in revenge for Italy overthrowing fascist dictator Benito Mussolini and joining the Allies, German troops flooded the Pontine marshes outside of Rome to trigger a malaria epidemic. Only following extensive campaigns of draining wetlands and spraying for mosquitoes was the disease finally eradicated from the continent. But while these surprising advantages of sickle cell trait might benefit the human population as a whole, they are little comfort to those who must live with the full-blown disease. Treatment for sickle cell disease varies widely, ranging from simple supportive care to cutting-edge gene therapies. For example, more minor vaso-occlusive sickling crises are usually treated with anti-inflammatory and pain-killing drugs, while increased susceptibility to pneumococcal infection is managed by vaccination and the prophylactic administration of antibiotics like penicillin between the ages of 4 months and 5 years. Other preventative measures include encouraging high fluid intake, avoiding crisis-triggering conditions like cold, stress, exertion, and high altitudes, and supplementing the diet with folate, aka vitamin B9, which encourages the production of more red blood cells. More active treatments include blood transfusions. This can take several forms, from a simple top-up to introduce more healthy red blood cells to exchange transfusion, in which the patient's blood is drawn out and sickle cells filtered out before being returned. Another common treatment involves the drug hydroxycarbamide, aka hydroxyurea. Originally a chemotherapy drug, the use of hydroxyurea to treat sickle cell disease was pioneered in the late 60s by hematologist Dr. Samuel Sharash at Johns Hopkins University. The drug triggers the patient's bone marrow to produce more fetal or F hemoglobin, which usually disappears from the body within 1 year after birth. F hemoglobin is more resistant to sickling than adult or A hemoglobin, helping to reduce the number of sickle cells in the bloodstream and the frequency of crises and other symptoms. It also stimulates the release of nitric oxide, a hormone that triggers vasodilation and improves blood flow. Another medication, voxelotor or oxbrighter, increases the affinity between hemoglobin and oxygen, helping to prevent polymerization and sickling. But while government approval for the drug was fast-tracked in 2019, by 2024 it was determined that the harmful side effects outweighed the benefits, and its manufacturer, Pfizer, voluntarily withdrew it from the market. However, in 2019, another promising drug, the strangely named crizanlizumab, was approved for use. This is a monoclonal antibody that binds to the adhesion protein P-selectin in red blood cells and reduces the risk of sickling. More sophisticated treatments currently being studied involve the use of stem cells. This involves destroying the patient's original bone marrow cells with chemotherapy drugs or radiation and replacing them with new stem cells lacking the defective HBB gene. And in 2024, 12-year-old Kendrick Cromer of Washington, D.C. became the first person in history to be treated for sickle cell disease using gene therapy. The procedure, performed by a team led by Dr. Andrew Campbell at Children's National Hospital, involved harvesting Cromer's own bone marrow cells and using CRISPR gene editing technology to correct the mutant HBB genes. Cromer's bone marrow was then destroyed using chemotherapy, and the edited cells reintroduced and allowed to recolonize the bone, completely curing him of his sickle cell disease. Another gene therapy under investigation involves editing the BCL11A gene, which normally inhibits the production of F hemoglobin in adults. This effectively replicates the effects of hydroxyurea, but without the need to take medication every day. Given the severe health risks associated with sickle cell disease, treatments must be started as soon as possible. For this reason, parents who carry sickle trait are encouraged to have their children screened for the disease. This is done in the womb via amniocentesis, the drawing of a small sample of amniotic fluid, or shortly after birth via a routine heel prick test. The newborn's blood is then tested for sickle cells via a number of procedures, including simple microscopic examination, electrophoresis, and chromatography. Prenatal screening, however, comes with significant ethical baggage, as parents may, if the option is available, choose to abort a fetus which tests positive for the disease. Indeed, around the world, sufferers of sickle cell disease are subject to significant stigma and discrimination. For example, in many parts of Africa, such as rural Uganda, the disease is seen as a punishment from God or the result of witchcraft, while the mothers of those with sickle cell disease are often condemned as irresponsible, and they're shamed for choosing to bring such afflicted children into the world. In the West, the long-held association between sickle cell disease and race has also had profound effects. In the United States and United Kingdom, over 90% of people with the disease are black, and as a result, it has historically been less well studied than other conditions, and sufferers have faced numerous barriers to accessing necessary care. However, the situation is steadily improving, and new developments in gene therapy promise to ease the suffering of millions or even cure them completely. Thus, in spite of the surprising evolutionary advantage this strange genetic hitchhiker has granted us over the last 7 millennia, the agony of sickle cell disease may soon be consigned to the dustbin of history.